recording this computer. Okay, cool. Hello, everyone. We're on our first question. Hello, YouTube. We're on our first question. We're just discussing question 8 here on ChemPhys, and we're wondering why or what would happen if the proton gradient was no longer coupled to ATP synthesis, right? And here in the question it says uncoupling protein, mitochondrial membrane. Okay, uncoupling, that's just a term found in biochemistry. Basically, what uncoupling means is that an action is not going to be coupled with something else, and what coupling is the use of a very thermodynamic reaction to get a non-thermodynamic reaction to occur. So let's say we had like this ball on the hill, right? This ball on the hill is going to roll down and is going to do its thing spontaneously. However, if we had a ball at the base of the hill trying to go up, is that thing going to happen spontaneously? No, it's got to be coupled with something that's spontaneous in order for it to happen. So in our body, it's ATP that is going to be driving this reaction. The hydrolysis of one of the phosphates from ATP is going to give this thing enough energy to go all the way up, right? So when it says uncoupling, that just means that, okay, ATP synthesis is no longer being done because the protons that are going down from the electron transport chain are not being coupled with ATP synthase. And when it's not being coupled with ATP synthase, you can't produce ATP. Okay, so let's go through the options here. So the electron transport chain. Okay, what is the electron transport chain exactly? Well, oh, snaps. Well, the electron transport chain is the movement of protons from the mitochondrial matrix to the inner mitochondrial space through the use of electron carriers, right? We have a bunch of electron carriers here. We have something in the middle, and then we have ATP synthesis, right? ATP synthase. So the electron transport carrier uses electron transport molecules like NADH, takes those electrons, moves it down this electron transport chain, and then eventually couples it with O2 to create H2O. Where does the hydrogens come from, you might ask me? Well, the hydrogens comes from the protons that are being moved up by the electrons being transported down this electron transport chain. Remember, electrons have negative charges. All right, so these negative charges are going to move down. They're going to pull those protons up, creating a gradient, right? A gradient means that there's a bunch of stuff in one area and not the other. There's a bunch of protons, aka H pluses, here in this inner mitochondrial in the inner mitochondrial membrane space, rather than the mitochondrial matrix. Okay, so this question is telling us, hey, what if this proton, instead of it coming down and kind of like helping ATP synthase create ATP and H2O, what happens if this is uncoupled? Well, will the movement of electrons necessarily be affected? Not really. Like, these electrons are kind of doing their thing, right? The only thing that happens is the NADH loses one of its electrons, and after that, it gets shuttled down all the way to H2O. Okay, so that's not affected. So what's going to happen? Is NAD plus reduction going to happen? No, NAD plus is actually, NADH is what's getting oxidized. Our NAD plus reduction, that's going to happen in either glycolysis and the PDC, the pyruvate dehydrogenase complex, or in the Krebs cycle, right? This wouldn't be happening in the electron transport chain section. Would the Krebs cycle be affected? Not really. We want to stick to this, like this proton gradient. The proton gradient happens in the electron transport chain. Okay, so can anyone tell me, what is oxidative phosphorylation? So think about the name, phosphorylation. Basically, what oxidative phosphorylation is the phosphorylation of ADP to make ATP through oxidative processes, right? So the oxidative processes here that we're focusing on is just cellular respiration overall, right? Just the creation of electron carriers like NADH in order for those electrons to be shuttled through the electron transport chain, in order for us to squeeze in an ADP and a phosphate together to make an ATP. That's oxidative phosphorylation. Substrate level phosphorylation is when you have just a random molecule that has a phosphate on it, kind of donate its phosphate to ADP, creating ATP. One of those examples could be creatine phosphate. Creatine phosphate is just a store of phosphates in our body. Creatine gives ADP a phosphate whenever we get really tired and we exhaust our ATP. But okay, yeah, that would be the correct answer, oxidative phosphorylation, because oxidative phosphorylation, the creation of ATP from ADP and phosphate occurs here at this step. If this step isn't being run by protons, well then guess what? Oxidative phosphorylation, aka the production of ATP from the ETC, Not going to happen. Okay, any questions? Okay, if not, we can continue forward. Guys, remember, you guys voted on super in depth last week. So do you guys still want to do like the super in depth thing, or do you want to like more kind of like blow past them?
I feel like the easier ones we can kind of blow past them, maybe. I mean, I'm not sure if everyone else feels the same way. I want to
That
speak.
that's what that's what I'm
Yeah, I
saying.
agree.
I don't know. I don't know what you guys consider easy.
Like, I mean, I feel as though you would kind of like the way that you classified it. I guess it would be okay. I mean, like,
All
what was the
right,
other one for the next easy peasy one?
this one
I can go through easy.
it's
You know, we don't have to go in depth. The concepts like what's the powerhouse of the cell? You know,
okay. This one's easy peasy squeezing lemons. So it says the structure of positive fructose is shown below. How many chiral centers does positive fructose contain? So positive, if it's throwing on positive on there, it's just trying to talk about optic ability. Remember, you guys, for you to have optic activation ability, like to be able to rotate plane polarized light, you have to be optically active. To be optically active, you have to be chiral. So this means that this molecule here is going to have chiral centers. Okay, so in order to designate what things have chiral centers, you need to make sure you're sp3 hybridized, which means that this kind of double bond here is not going to cut it, right? Because this double bond here, this carbon now has an sp2 hybridization, not an sp3 hybridization. Looking for an sp3 hybridized molecule, or sorry, carbon center with 4 different substituents. We have that in this carbon here as well, and this carbon here as well, right? Because this carbon has an OH, an H, and then a CH2O. This carbon has an OH, an H, carbon that leads to a carbonyl, and then a carbon that doesn't lead to a carbonyl. Okay, cool. This one's different. How about these? And hydrogen and OH, carbon that's attached to a carbon that's a part of the carbonyl group, and then a carbon that's not attached to a carbonyl group, making them sufficiently different, meaning these are chiral centers. In total, we have 3 chiral centers total. Is that kind of the speed you guys want? Is that good? That speed? Okay, cool. I don't want you guys to be like, whoa, he's rushing things. No bueno. Yep, that's good. Okay. If everyone agrees, then we can keep it at that pace. But I mean, that was also like really easy. But okay, let's continue. That's the thing. I don't want someone to be lost, but at the same time, I guess you can't sacrifice the sheep for the herd or something, if that's the thing. Okay. In reaction 1, the compound glutamine synthase serves to, okay, cool. So I kind of like read this question first. I didn't really think I needed to know what's it called? I don't have to read this entire passage in order to solve this question, right? Just asking about what glutamine synthase serves to do in reaction 1. Okay, in reaction 1, we have ATP, we have glutamate. Remember, glutamate is an amino acid that's acidic. It's just the ion version of it. And then we have here ammonia, and then we have the production of ADP, a phosphate, and a glutamine. Oh, okay, what does ATP and in the reaction side, an ADP plus phosphate mean? Okay, so it means we kind of like needed energy for a reaction to occur. Here we're starting off with glutamate and NH3, and then we're ending up with glutamine. Glutamine is basically just glutamate, except the OH group is just replaced by a nitrogen group. So you make an amide. So what I'm guessing here is just that the glutamate, which kind of looks like this, OH here, or if it's glutamate, then it has the ion, like so. I'm not going to draw the entire amino acid, just imagine the backbone over here. And then the NH3, the ammonia, kind of came in, replaced this OH, and then we have our handy dandy amide in the form of glutamine, NH3. Right? Okay, so this is what, or actually NH2. Don't have a charge on that guy. There's no charge on glutamate's amide. Okay, so that's what happened. Cool. So, all right, that's the reaction. It's asking about synthetase. What does the ASE mean? Can anyone tell me?
It's an enzyme.
We're working with an enzyme. And what does an enzyme do? It affects things kinetically. It doesn't affect things thermodynamically. So that's what we're going to have to look out for. Increase in Gibbs free energy. Is Gibbs free energy kinetic or thermodynamic?
Thermodynamic.
Yes, it's thermodynamic. Thermodynamic. How about activation energy? What's that?
Kinetic.
Anyone?
okay.
Well, yes. But why? Well, if you guys look at a reaction, let's say this is the reaction coordinate right here, right? Here we have reactants and here we have products. The distance between these two right here, that's thermodynamics, right? Just the difference in the Gibbs free energy between the reactants and the products. Everything else is pretty much just kind of related to the kinetics, right? All this energy over here on top that's not involved in just the net difference between the reactants and product, that's kinetic stuff, right? Specifically, this kind of hump right here, this total energy, that's activation energy. And activation energy is something that controls the kinetics. Because if you need a reaction that requires a bunch of energy, guess what? That reaction is going to happen slowly because somebody's got to gather that energy. But if you have an energy that's like that, if you have a process that has really low activation energy, well, then you're going to it's going to be faster because it's going to happen more rapidly. It's going to happen more often. What an enzyme does, aka glutamine synthetase, is that it just decreases the activation energy of a process. How? By making the transition state very stable, right? We talked about how there's a transition state in the process when you're making reactants from products, right? The reactants turn into kind of like this kind of like, you can think about it as a glob where it doesn't know what it is yet, kind of like the analogy of having, cut up a produce here and then you put it into the frying pan. It's like just high energy. You take it out and you serve it at like a lower energy, right, as food. So here, just like you have a glob, there's a transition state. So basically, what the enzyme does, it makes that glob like just be stable. Remember, the more stable you are, the lower energy you are. Okay, so that's what an enzyme does, right? So decrease the activation energy. Yes, that's what something that an enzyme does. How about decrease the entropy of the reactants? That's not what it does. The entropy of the reactants is just how folded it is or how unfolded it is, just kind of how nuts the molecule is. The enzyme does act to kind of like capture molecules and like fuse them together so they can be in the same spot and like occur rapidly, but that's not increasing the entropy of the reactants itself. That's just that's just like decreasing the movement of it. So that's more what's it called, like the movement of it, and that movement of it is not entropy, so C would be wrong. And then shift the equilibrium to the right. The only thing that can determine equilibrium is your thermodynamics, right? How spontaneous something is, right? If something's really spontaneous, then you're going to have more equilibrium to the right. Remember, this is our arrow. We have reactants, products. If you have more equilibrium towards the right, so let's say equilibrium lies right here, then you have a spontaneous. Oh, we have R and R. Right here in this direction, then we have a lot of product. If the equilibrium is over here, then we're going to have a lot of reactant. The enzyme is not going to touch upon that. Only your Gibbs free energy and temperature. That's the only thing that can influence those two things. Okay, how is the speed for that one? I feel like it would be better if you just explain why your answer is correct or incorrect for the harder questions. Was that speed good? Speed perfect. Cool. Oh, okay. Yes. Okay, cool. Because I don't want you guys, because like if you guys like zone out for a second, I talk fast, so like you're gonna like lose what happens. Okay, yes, guys, let's do this. All right, question 22 on ChemPhys. I'm pretty sure if we go at this rate, we're going to finish all these questions. And these are just the easy peasy questions. I have a different PowerPoint for the less peasy and the non-peasy questions. Okay, let's see. Okay, so I'm shuffling through my full length and I find this question here. In its interaction with the magnesium ion shown in figure 1, the ASN, okay, so whenever you see like a three-letter word and then a number, you're dealing with an amino acid. Basically, it's just an abbreviated amino acid and then the location of that amino acid on the whatever it is, a protein, it could be a protein, aka an enzyme, a transporter, whatever it is. Okay, it says, in the interaction with the magnesium ion shown in figure 1, ASN acts in which of the following ways? Okay, so first off, when I'm thinking magnesium ion, what kind of charge? Oh, again, I gave it away. What kind of charge am I thinking about? Well, this is a metal, right? So it's most likely going to have a positive charge. Okay, I'm just going to keep that in mind. Okay, so it's asking us to focus in on ASN 581. That's this sucker right here. Okay, so right here we have a nitrogen group, right? Because it's an amide. We have a nitrogen group and a hydrogen. You guys remember what a nitrogen group and a hydrogen can do? They can act as a hydrogen bonding pair, right? Like any lone pairs on FON can be a hydrogen bond donor, meaning that they can have the lone pairs nice and chilling, or they can, actually, sorry, that's a hydrogen bond acceptor, or the hydrogen bond donor, which is a hydrogen covalently bonded to one of these three atoms, right? So here we have nitrogen and we have a hydrogen here. So we have potential for hydrogen bonding. That's what's going through my head. Say ASN, right? Or ASP, yeah, ASN. Okay, so just look, let's follow the reaction. We don't need to read anything else, right? Just look at this reaction here. Okay, so we have the magnesium. It's attached to something that has 3 phosphates here. Recognize these structures, right? This is a phosphate group, this is a phosphate group, this is a phosphate group right here. And then we have a ribose sugar, and then it's kind of like a thing that has a bunch of nitrogens. Guys, what does that scream to you? What does that tell you that this structure is right here?
Is that ATP?
That's what ATP looks like. This is ATP, like 3 phosphates and blah, blah, blah. And then we have a magnesium ion doing this thing over here. Okay, so you have the ASN. Okay, what does it seem to be doing here? This straight line means that there's going to be a covalent bond forming. A dashed line means that there's just going to be a sort of hydrogen bonding occurring. So here, actually, this is just a reaction arrow, just pointing where things are moving. Actually, just the movement of electrons, this arrow right here, and then the dotted lines are just forces between them. It could be intermolecular in this case, or they can be intramolecular in a different case. Okay, here we have the kind of connection between these two molecules, the Mg, this atom, and over here the amide of ASN 581. Okay, so it seems like they're kind of bonded there. What else is it doing? Here if we go on further on, it just keeps holding on to the magnesium ion and keeps holding it on, and then it eventually lets it go. Okay, so what can that be, right? So you go down here, a Lewis acid. Well, it's asking us what the ASN 58 is doing, 581 is doing. So is it acting as amino acid? I mean, check this out. It says, and remember, guys, from Orgo, the reaction arrow, the thing that's at the base of the arrow, so the thing that's not has the pointer on it, that's where the lone pairs are, the things that you're transporting, right? It could be a double bond, it could be a lone pair. That's like one of the first things that you guys should recognize. Like, for example, if we had like this structure right here, the double bond would be going in this direction, right? If you had if you had this kind of situation over here, then the lone pairs here, the arrow would be drawn like this. So when you have a arrow drawn like that, that means that whatever's over here, that's a lone pair. You guys remember, nitrogen has, if it's not charged, or if it doesn't have a positive charge, then it has a lone pair, just not drawn. Okay, so is it acting as a Lewis acid? What's something that's a Lewis acid? You guys remember Lewis loss or Lewis lone pair, right? So it's going to be something related to electrons. So is it acting as a Lewis acid? A Lewis acid is something that accepts lone pairs. Is it accepting a lone pair anywhere? No. So we can cross that one out. Is it giving lone pairs? Well, let's look at this. Magnesium is something that has a positive charge, right? What is something that have positive charges? What do they want? They want something that has negative charges. Okay, so we're going to have the lone pairs here on this amide kind of be shared with the magnesium. We're going to have this bond over here, and what this what this thing is showing us is that the ASN, it really acts to just hold ATP in place, right? That's the job of ASN, right? If it's being created back at the end and the magnesium just kind of is still there, like there's no net change in ASN, well, guess what? That thing is just kind of there to hold things, right? Okay, so that would be the correct answer. A Lewis base. Did it lose a proton? Did it gain a proton? No. So it's not a Bronsted acid or Bronsted base. Okay, so that would be question 22. All right, question 25. Same passage. All right, let's continue. All right, so it says, according to the mechanism shown in figure 1, what would figure 1, would the function of the BRAF kinase domain be eliminated by the substitution of glue for ASP? Okay, so we're going to look for the BRAF kinase domain. What does that mean? So I guess we're going to have to read the passage a little bit. But basically what we have here is the substitution of GLUT for the ASP57. So glutamine is glutamine is something that's acidic, is going to be replacing ASP. Aspartic acid is something else that's acidic. Typically right here, I would kind of just like stop and answer it, because if you're having the exchangement of two acids, they're pretty much going to do the same thing, right? The only difference between glutamine and aspartic acid is like one carbon group, right? So they're basically the same thing. They're interchangeable. The bases, for the most part, like the basic amino acids, they're also interchangeable. So I would just look at the no's. I would cross out the yeses because there's nothing really significant that I'll change from the glutamine to the aspartic acid. So something very specific after the no is what makes me want to like try to refer back to the passage if I need to. Here's look at A. Glue can bind to the divalent metal ion chelated by ATP. No, glue can activate the nucleophile shown in step 2. I'm not sure what is going on here, so I'm gonna have to refer back to it. Says the figure 1, what are the function of BRAF kinase domain? Okay, so not really sure what BRAF is. I'm gonna have to read it, the description of the figure. BRAF catalase phosphorylation of serine 309 residue at the gamma phosphate group and ATP. So is this whole thing BRAF? Okay, let me just confirm. All right, here BRAF is a serine 309 protein kinase that catalyzes the transfer of phosphate group from ATP to serine. Okay, we have ATP here. I see the movement of phosphate Y because there's no longer a phosphate group going on here. So I'm guessing that BRAF is just this whole thing, right? Okay, cool. This BRAF is this whole thing, and it's asking us, would the ASP 576 would it, well, with this kind of domain, this catalytic domain, would it change if glutamine was put in there? Well, what is the function of ASP? The function of ASP is to snatch away a hydrogen here, and then after that, it just kind of hydrogen bonds with the ASN. Okay, can glutamine do that? Well, yeah. The difference between aspartic acid and glutamine and glutaric acid really isn't that much. So okay, let's look at the second part of the question statement. It says glutamine can activate the nucleophile shown in step 2. Okay, so what's the nucleophile here? It's this thing right here. Remember, what is a nucleophile? It's something that can be negatively charged, something that has lone pairs, or that's something that has like a double bond, for example, right? So what is the thing that's activated here? Let's just look at specifically at ASP. ASP is removing a proton from this oxygen molecule. Here, I'm guessing it's 309 or serine. It removes that hydrogen from 309 or serine, and then it snatches up, and then this molecule right here, because it has a negative charge, is a really good nucleophile, right? If it has a negative charge and it has lone pairs, it's going to be a super nice, strong nucleophile. It's going to snatch away one of those phosphates and phosphorylated stuff. Okay, can can glutaric acid do that? Yes, it can, right? It can activate the nucleophile, because if aspartic acid can do it, glutaric acid can do it as well. And that's what's happening, right? The nucleophile is being activated with the removal of the hydrogen. Let's look at A. Glutamine can bind to the divalent metal ion chelated by ATP. Well, is aspartic binding to the divalent ion? Like, is it binding to this thing? Is it really interacting with it? No. Okay, so A is incorrect. So 25 would be B. Okay, moving on. All right, so same kind of passage. All right, moving here. PLX 4720 and PLX 4032 would be expected to bind with greatest affinity to any enzyme active site containing a large number of which of the following residues. Basically, you're just looking for the thing that's different, right? Because if they're going to present to you a bunch of amino acids, you basically just have to choose the amino acid or the group of amino acids that are different from the other ones. Just kind of look at the kind of situation that's going on here. V and L, those are both non-polar. D, that's an acidic. This is polar. This is acidic. This is basic. This is what's it called? Basic, and this is polar, right? The only one that's different is A. So I would choose A right off the bat, but let's just confirm, right? With the passage, we refer back to the passage. It states that PLX is, where does PLX start? Over here it says PLX, and it states that PLX is going to be binding in a non-polar region. Okay, that confirms our theory, right? So V and L, the things that are non-polar, if it's a non-polar region, we're going to want what? Non-polar amino acids, which is V and L, valine and leucine. Both of them are non-polar. The rest of them don't fit the description, right? They're either polar or they're acidic or basic. Remember, they're acidic or basic, they can like have the movement of protons, which means that they can have a charge potentially, and a charge is the opposite of non-polar, right? Okay, so that's 26. All right, let's continue forward. Let's march on out. All right, so here we got 31. The objective is to finish all these questions. I don't know if you guys noticed, but every week I've been getting like the most amount of questions out of your 3 instructors. I thought Orgo and Biochem was just kind of like, who cares? But TPR really cares about those guys. All right, so let's see, question 31. All right, the trimerization. Okay, so right off the bat, trimer. Can anyone tell me what trimer kind of tells you what's going on?
Isn't that like multiple subunits?
Specifically 3, right? Trimer, 3.
Mhm.
What happened?
No, I was just saying, yeah. Yeah.
Oh, sorry. Okay. I thought you were just like, I thought you had a question. All right. So trimerization just means a process, right? So trimerization is just a process of making things that are three, right? Okay. So three making process of adenovirus, protein fiber, monomers is an example of what type of protein structure. Okay. Right off the bat, if we're talking about monomers and we're talking about protein, we're talking about trimerization. I know that something's going to be, related to protein structure, right? And I mean, they lay it out here perfectly with the answer choices. It's primary, secondary, tertiary, quaternary, right? So here I've listed in the PowerPoint in case you wanted a brief reminder. Primary is just the amino acid chain. Secondary, you have hydrogen bonding, so we do have some folding. Tertiary is hydrophobic, hydrophilic interactions. It'd be like van der Waals, whatever it is, like your polar group, your dipole moments, whatever it happens to be. And the quaternary is just when you have like two different subunits. Let's say like subunit A. Oh, it's not letting me draw. I say subunit A and subunit B, and those guys have a bond for whatever reason. It could be like a disulfide bridge. Typically, that's what you can be finding with like bridges. Between like subunits can be a disulfide bridge with cytosine. Cytosine has a sulfur group, a disulfide bond, just two sulfurs connecting to each other. That'll be typically your quaternary structure, right? You have bonds between separate. Oh, I didn't finish this. Separate chains, right? Separate change. And then here it says trimerization and monomers. Okay, yeah, that's basically the answer. It's quaternary because we have three things binding, right? Three different monomers binding. It's really just the recognition of vocabulary, right? Okay, that's 31. And guys, remember these PowerPoints are up on Sakai, so I see some people rushing to break their fingers. No need to take your notes at breakneck speeds. This information is available to you guys. Okay, so here we have intermediate X. The question is asking intermediate X formed in experiment 1. Okay, so if it's talking about an experiment or a reaction, I'm gonna just float towards what the experiment is, see if I can derive the information from it without you needing the passage. If I can, I'll do it. If I can't, then I'll just move on. So, okay, so it's telling me intermediate X formed by an experiment 1 is, okay, so let's look at intermediate X. Let's look at this reaction here. Okay, we have a ketone group, we have a base. Guys, remember what can a base do to a ketone group? What's acidic about remember if we're introducing a base, it means that we're trying to exploit something that's acidic, right? So what's acidic about a ketone group?
Is it the alpha carbon?
Yes, the alpha carbon is acidic, right? The more particularly the alpha hydrogen is going to be easily popped off. The reason why the alpha hydrogen can be easily popped off. So remember, alpha just means one carbon away from whatever it is that you're talking about, your reference point. In this case, the carbonyl group is our reference point. So this one would be alpha, this carbon here, this one would be beta, and this one would be et cetera, whatever, right? So these alpha hydrogens, they're acidic, and the reason why they're acidic is if you were to plop one of them off, right? Let's say we have a base here, and the base snatched one of those hydrogens and it turned into a water molecule, then well, who's more electronegative, the carbon or the hydrogen? The carbon, right? So the carbon is going to keep those electrons. All right, so if it's going to keep those electrons, we're going to have a carb anion. Okay, typically a carb anion, guys, is a carb anion reactive? Is it stable?
It's unstable.
Yes, it's unstable, right? It's something that it's a reactive species. It doesn't really like to be there. So what we can do to kind of stabilize this is we can do resonance stabilization, right? So typically a carb anion, bad news, right? That's because like a random, let's say a propane. Oh, that's a butane. Say we have a propane, right? This thing's not acidic. It's like this little molecule is not going to be randomly donating its protons. However, in this case, we have a carbon group donating its proton because this lone pair, just remember with the reaction arrows, this lone pair can go form a double bond here, and then the double bond here can move its electrons to the oxygen, forming this structure over here, right? So whenever we have this, it's just the movement of this negative charge. Now it's moving on to the oxygen. Whenever we have this kind of scenario where we can have a carb anion right next to a carbonyl group, this is called the enolate, right? They're enolates of each other. And in one case, we have the carb anion. In the other case, we have the oxygen species having the negative charge on it, right? These are our enolates. Okay, so this is basically what's going to happen in this experiment, right? This is enolates, like, okay, where have you heard enolates before? This is definitely not your first introduction to enolates. Enolates happen in something called the aldol reaction, right? Something along the lines of the alpha carbon of a ketone group is going to become a strong nucleophile. Why is it going to become a nucleophile? Because look at this negative charge here, right? This negative charge is going to make it a good nucleophile, especially if it's a carbon, right? It's a really reactive species. Well, basically this carbonyl group, this enolate, right? This negative charge, what it's going to do is it's going to tackle the partial positively charged carbonyl carbon, and then it's going to attach and form a molecule, right? That's an aldol an aldol reaction, just a basic aldol reaction. But here's just asking what the intermediate X is, right? So when we have the OH pull off our one of our hydrogen, one of the alpha hydrogens, we're going to have this enolates. Okay, so that's what the intermediate X is here. We have that a CH3 ion, CH3 iodine is interacting later on, but we're not focused on that, just the intermediate X. Okay, so what is the intermediate X telling us? Is it an alcohol? Well, is an oxygen by itself, is that an alcohol? Like this enolate right here with the negative charge on the oxygen, is that an alcohol? No, it's missing hydrogen. Okay, we can cross that one out. Is it an alkene? Well, no, right? I mean, an alkene is here, it's there, but this is a very specific alkene. It's sort of as an oxidative alkene. So in terms of its specificity, now I kind of took issue with this question because an alkene is formed, like an alkene is just a double bond between carbons. So that is formed. So I was like, man, I don't know, like that should have been correct. But at the same time, resonance stabilized is another thing that can define, what's it called? What's going on here, right? So the ion from the carbon anion can shift over, form a double bond between the two carbons, and the double bond between the carbon and the oxygen can go up and move on to the oxygen, causing resonance stabilization. C would be the correct answer. A carb cation, no, the carb cation wouldn't be forming because the base is removing a hydrogen and leaving behind electrons. It's not taking the electrons as well. So a carb cation, that's just a positively charged carbon group. If you have a positively charged carbon group, what you have is a something that's electron deficient. That's not what's going on in this reaction. Okay, any questions about 35?
Yeah, I have a quick question.
Yes.
So let's say that like the original like part, like where the ketone is, like what if there was two ketones on there? Like how would you know where the hydrogen's being taken off from?
Well, that is more of a kind of like organic chemistry Rutgers kind of question. Like for the MCAT, they're not going to go that deep into it, but you just
Okay.
if that ever happens, you just have to look at the resonance stability, right? And like the stability of your carb anion. So remember what we said, how to categorize if something is a strong acid. What do we do, Alexis?
Strong acid has
What is its conjugate base?
is it a weak base?
Yeah, a weak base, aka it's stable, right? So
Okay.
the more stable the resulting conjugate base is, so this is an acid, right? And then remove the hydrogen and it turns into a conjugate base. If this conjugate base is super stable, that means that this proton is readily to hop off, right? But if this thing is unstable, the reason why we have in the weak acids, the reason why we have partial deprotonization means that there's not complete kind of diffusion between the proton and the parent compound, is because this thing is so reactive that it kind of just snatches the hydrogen back, right? So whenever we have strong acids, we have complete dissociation. Strong weak acids, we have lesser dissociation. How can we determine if something is going to be dissociating or not? How stable the conjugate base is, right? So if we were to have like this molecule right here, for example, which one of these, A, C, or B, which proton do you think would be snatched out first?
Would it be B?
Yeah, because B, if we were to form the carb anion over here, what would happen is that we have resonance stabilization in this direction, but also resonance in that direction. If we were to have some sort of inductive effect, right? If we had, remember we talked about the fluorines, how the fluorines can help us out with the kind of control of negative charge, right? Let's say we had for
Trifluorine.
Yeah, I was going to make it more complicated, but yeah, you can just do this, right? Okay, which one will come out first? This one, or let's just say that these were just methyl groups, right? Just methyl groups.
Mm-hmm.
Which one come out first, A or B?
It would be B. Or no, sorry, A. I read that wrong.
I was like, Alexis, you were like so confident.
A, yeah.
Yes, because the negative charge resulting from the proton A proton moving the fluorines, they're electron withdrawing groups, right? Because they're electronegative. So they're going to be pulling that negative charge, making it more stable as it's being, that negative charge is being dissipated throughout the molecule. Rather over here, these methyl groups are electron donating groups for the sake of the MCAT, and they're going to be pushing electron charge towards the negative. Remember, two negatives together, those are not mixing well, right? So it's going to be unstable. So that's how you can categorize it.
Okay, thank you.
Mm-hmm. Okay, any other questions? Okay, cool. So that's question 35. All right, let's see what else we got next. I'm pretty sure we can finish all of these by the time. Oh, I'm gonna take that back. All right, I'm not sure. All right, let's continue going. Okay, here we have 36. Man, I'm parched. Kind of sound like what is his name? I forgot. Okay, so okay, question 36. We have here the same passage. It says competing reactions in experiment 1 are responsible for the product mixture. All the following reactions contribute to this product mixture except, okay, so let's look at experiment 1. In experiment 1, here it said that we have some sort of mixture of products. Okay. So what can happen here, right? So let's see here. We have the alpha hydrogen being plopped off from the by the use of this base. We have the intermediate X, which is basically just an enolate, right? Either we have the oxygen with our negative charge and a double bond here, or we have our oxygen nice and fully without a charge, or we have a carb anion over here, right? We have this as our intermediate X. Here we have CH3I. CH3I looks like so. The iodine is very electronegative. It likes to have its electrons. CH3 is kind of indifferent to it, right? So if we were to look at these two structures, remember they introduce, and this arrow is basically saying that they're introducing CH3I to this intermediate. If we're looking at this kind of thing going on here, well, what's going to happen, right? So here we have a negative charge, and here we have a negative charge. And here we have iodine that's partial negatively charged, right? So basically what we've been looking at is a nucleophilic substitution. The reason being is that if these things have negative charges, especially for this carb anion, right? This carb anion is very reactive. This iodine, it's a good leaving group because if remember what a good leaving group is, it's either it's resonance stabilized or it's a weak base. And iodine is definitely a weak base. Why? Because its conjugate acid is a strong acid. This is the conjugate acid of iodine, right? So an iodine by itself, just an iodine like this, like an iodine ion, is a weak base, right? It's not going to be, it's going to be chilling pretty much. So what's going to happen is we're going to have a mixture of products because the iodine can plop off, right? This nucleophilic carbon can, now that the CH3 is missing, let's say the iodine's here. Now that the CH3 is missing, well, just to I guess be consistent with everything. What kind of reaction would be happening here? Would it be a SN2 reaction or an SN1 reaction? Can anyone tell me?
Would it be SN2?
Mm-hmm. SN2 because look at this structure right here, right? It's just a methyl group, right? So this is something that's really not highly substituted at all. And remember that substitution, SN2 reactions, they like things that are not substituted. So basically this nucleophile is going to come in through here, bind to our CH3. We're going to have a pentavalent transition state for a second, and then the iodine pops off, right? So the reason why we have a mixture of products is because this oxygen species here can also act as a nucleophile and come in to attack. Or what can happen is the aldol reaction, right? Remember what the aldol reaction is basically this negative carb anion tackling the partial positively charged carbonyl carbon of a different version of this species, right? So that's something that else can happen. So that's the mixture of products that can potentially occur, right? Okay. All the following reactions contribute to the product mixture except, okay, deprotonation. Did that happen? Yes, our OH kind of snatched away an alpha hydrogen, right? Okay, cool. Okay, an aldol reaction. Can that happen? Yes, I didn't know aldol reactions before, so that's why I chose it. But basically an aldol reaction can happen because we have two ketones and we have base. Whenever you have two ketones and a base and there is an alpha hydrogen, like let's say we had these two ketones, right? And this is a benzene group and this is a benzene group. Let's say like this is in our mixture, right? Can we have an aldol reaction if we had two of these? Can anyone tell me? Hello. Okay, I'm guessing you guys know. So yes, if you don't have an alpha hydrogen, let's say like an alpha hydrogen is like right here, right? Like this is your alpha hydrogen. If you don't have an alpha hydrogen, well, guess what? A base won't be able to remove that hydrogen, and you won't be able to form your carb anion. The carb anion is going to be the nucleophilic attack for the aldol reaction. So here we have two benzene groups. Let me draw this better. So, or let me just draw a portion of. Okay, let me just not be lazy. This is your benzene, right? So here you have a hydrogen, you have a hydrogen, you have a hydrogen. But is there a hydrogen on this molecule? No, because there's already 4 bonds on this carbon. So you're not going to have any alpha hydrogen, right? So if you have no alpha hydrogen, you're not going to have aldol reaction. Okay, but here we do have alpha hydrogen, so you can have an aldol reaction. Okay, nucleophilic substitution. Yes, that's what we talked about, right? What's an SN2 reaction? SN2 reactions are a substitution reaction using a nucleophile in either a mono molecular or a bimolecular kind of way, right? So that's what we have there. So nucleophilic substitution definitely occurs. A hydride shift. Does a hydride shift occur? No, a hydride shift only occurs typically where you're going to see a hydride shift is in SN1 reactions, right? So basically a hydride shift is, let's say we have this structure here, and for whatever reason, we have a positive charge right there, right? Well, guess what? Remember like the electron, what's it called? Electron withdrawing groups and the electron donating groups and all that, like how to stabilize a carb cation. So which one would be the most stable carb cation from these 3 species? A, B, or C? Which one would be the most stable?
B.
Yeah, B, because there's a lot of electron donating groups on this carbon, right? It's a tertiary carbon. And so the negative charges are going to be pushing towards that positive charge, stabilizing it, right? Because they're oppositely charged. They're kind of the opposites of carbon anions. So whenever you have, let's say like an SN1 reaction, remember in the SN1 reaction, the leaving group just pops off and then you have a carbocation, right? Well, a hydride shift is going to occur whenever you have something that could potentially be more stable for the carbocation. So remember, this tertiary carbon has a hydrogen over here, right? So this molecule is going to be smart. It's like, hey man, like this carbocation right here on my secondary carbon, it's not cutting it for me. I want, oh, that kind of looks like an A. I want to move that thing to, oh, okay. All right, I can do this. Yes, a hydrogen. Basically, this hydrogen is just going to move itself. It's going to take its electrons and everything, move itself to over here. And now guess what? We have a carbocation in this tertiary carbon instead, right? So a hydride shift happens with carbocations or something that's positively charged, and it's not going to be happening for an SN2. It's going to be happening mostly for an SN1 reaction. Okay, so that's 36. Any questions? Nope. Okay, let's continue forward. Here we have 37. So you guys, is that good then? The what our plan is for next week, just like content review, first for an hour and then error analysis. Yeah, okay, I see some head shakes. Cool. Yep, okay, awesome. Yeah, because I don't want to take your Saturday away from you guys. So I want to, whatever you guys want is what I'm gonna do, pretty much. My boy? I don't know how to say it in Spanish. Just pretend I did it with a Spanish accent. Cool. Okay. All right, let's see, 37. In the alkylation step of experiment 2, so okay, the alkylation, what's an alkyl group? That's a carbon hydrogen group, right? This can be a methyl, could be an alkyl group, a propyl, or this is an ethyl actually, but if you were to like draw like this is an methyl, this is a ethyl, like these are alkyl groups, right? So alkylation is just the addition of a carbon group, right? Okay, it says, and the alkylation step of experiment 2, the carbon beta to the nitrogen act as, okay, so let's look at experiment 2 real quick. Okay, we have a ketone, we have a secondary nitrogen, and we have acid. Okay, I think I know what's going to happen. We're going to have the creation of an enamine, right? An enamine basically looks like this. look at the name in mean. Oh God, what does En mean typically? Can anyone tell me? In, like, what is this meaning of this? In organic chemistry, does anyone know? It means double bond. Oh.
Okay.
Sorry. And then an amine is just a nitrogen group attached to a carbon, right? So an amine looks like so. And you can have an amine formation whenever you have a secondary nitrogen. Here we have a secondary nitrogen, right, of two carbons attached to this nitrogen group, and then we have a ketone. With acid, you're going to form an amine, right? Basically, just the nitrogen group is going to be replacing this oxygen. So, like, if you want to know what the product is, it's basically right here, right? This thing is just like that, right? And then you have the double bond on the alpha hydrogen over here, right? It could be a double bond in this direction, it could be a double bond in this direction. And this particular molecule doesn't really matter because either direction is the same thing, right? So that's, we're gonna have an amine formation, right? Okay, so that's first things first. Well, when we have an amine formation, look at this situation that's going on over here, right? Over here we have the nitrogen group, and we have the I see something in the chat. Okay, so we have, let's draw our amine here. So we have a nitrogen, we have this structure over here, and you guys remember there's always a lone pair on nitrogen. There's no charge on it, that means there's a lone pair because it likes to have 3 bonds and it carries just a lone pair, right? Okay, so we, oh shit, messed up on that last one. Oh, like, you know what? I'm gonna just stop half-assing my drawings because I'm an adult who looks like a child. It's shaved, and I have a hydrogen here, double bond there. Okay, basically, we have what, the potential to have the removal of this hydrogen, and what can happen there is a carb anion, right? So actually, not sorry, I said that wrong. Not the removal of the hydrogen, the removal of the double bond, right? And the removal of the double bond looks like so, right? The lone pairs on the nitrogens go down like this, forming a double bond, and the removal of this double bond goes over here, and then we have a carb anion forming, right? What does this remind you of? This kind of reminds you of the enolates with the oxygen species, right? The nitrogen has a plus, and then we have a carb anion over here, and then we just happen to have a hydrogen like so, right? So can anyone tell me what this carb anion, this negative charge species, what can it potentially do? What can it act as? A file of what? What kind of file?
Nucleophile.
Yeah, it can act as a nucleophile, right? Because it has this negative charge here. It has a lone pairs it's able to donate, and guess what? It's a carbon group, which means that it's reactive OD, right? So okay, so that's what we can have with an amine, right? Typically, if you want to add a carbon group to a ketone, well, first what you're going to do is you're going to introduce it to a secondary amino group, and then you're going to form the enamine, and then you're going to just kind of like wait for the this double bond to like form the enolate version of it. So like the negative charge over here, due to just like, it's just resonance stable. Nothing kind of moved, right? It's just the resonance, like the movement of electrons, sure, but like nothing in terms of like atoms moved, only the electrons moved. And then after that, you're going to introduce it to whatever it is that you want to add, right? The alkylation step, right? So you're going to have, like, let's say in this case, we have a CH3. And this nucleophile is going to compete for the CH3. The iodine is happy to leave. It's a good leaving group. And then we're going to have alkylation, right? The result is going to be the, what's it called? Nitrogen is like so. What we're going to have here is the CH3 like this, and the hydrogen like this. And then after this, you know, this double bond doesn't have to be there anymore because it could just do, well, I mean, this positively, it will form a carb cation there at the bottom. So basically, once this forms, you're going to have, yeah, you're just going to add acid, so you can protonate your nitrogen and have it leave off. The mechanism is in the slides in the winter session. So you add the acid for the protonation step, and then the nitrogen is going to be basically washed out, and you're going to have your ketone form again. Okay, so the question, the alkylation step in experiment 2, the carbon beta to the nitrogen act as. Okay, so remember, beta is the thing that we're looking at. Alpha means that it's one carbon away. So it's like, we're not gonna be looking at these carbons, right? There's nothing really happening with these carbons. We'll be looking at the carbons over here. So, okay, this is the alpha hydrogen and this is the beta hydrogen. Well, guess what? Guys, can you tell me without looking at the answer choices, what was this carb anion? What was it just acting as?
Nucleophile.
Yeah, so that's the answer. Boom. A nucleophile due to a partial negative charge from resonance. Bam. And that's the answer. I didn't know that at the time. Yeah. So just guys, if there's a breach in your content, hey, it's all good. Just learn it, you know, and then you'll be set. All right. Any questions for 37? No. Okay, cool. Let's continue marching forward. We have question 38 next. All right. Here we have, if the students had used a propyl amine instead in experiment 2 in place of the pyrimidine, this is a pyrimidine right here. Guys, remember the list of molecules that I gave to you in one of the biochem, what's it called, PowerPoints, like kind of like just molecules cool to know. Whenever you see something like pyrimidine, this is something that's typically what you're going to be seeing for the alkylation step of a ketone, right? Whenever you're trying to use enamines to alkylate a ketone. So that would be something maybe good to know, right? But sometimes they'll tell you it, so it doesn't really matter or not, but like definitely revisit that PowerPoint and like learn those structures because they might pop out. I'm guessing some of you have already seen them in your test. Okay, so here it says, what would be the functional group that would be synthesized in the first step? Okay, was the first step? Okay, this top step. So basically it's saying a propyl amine. What is a propyl amine? We'll just look at the name. It's a propyl group attached to a nitrogen. Oh, snaps. And a propyl group is a 3-carbon structure. So 1, 2, 3. Okay, so what is this right here? It's an NH2 with a propyl group. Can anyone tell me, is this a primary or secondary amine?
Oh, it's primary.
Yep, it's a primary amine because there's only one. Oh, this nitrogen looks like it's part of the group. Because it's just one carbon attached to nitrogen, right? Well, if a secondary amine with a ketone washed with acid forms an amine, a primary amine washed with a ketone and acid is going to form an imine. An imine looks like so. Basically, your oxygen group is just replaced with a nitrogen, and that's all that happens, right? And then you have your thing like that. That's an imine. Kind of looks like an I. That's how I memorize it. An imine, right? Kind of looks like this kind of structure here looks like an I a little bit, not really. So that's an imine. And that's basically it. That's just, they're asking you, do you know your reaction? And you say either yes or no. And I happen, I'm pretty sure I guessed on this, but I got it right. Okay, any questions for 38? Nope.
Wait, so I guess it's great to know that typically if you have like a secondary amine or amino group, that you're going to usually form an enamine, and a primary is going to be an imine.
Yeah.
Okay, cool.
I said that. I hope you guys heard when I said that. But yes, thank you for the recap, Kevin. He's such a good student. He's just like, he's looking out for you guys too. Whenever he says that, he wants you guys to remember too. If that's what you're going for, thank you, Kevin. If not, then okay. All right, so let's continue forward. 44. Yes. Okay, cool. Freestanding question straight up. The tertiary structure of a hydrophilic sodium hydrogen transport channel would best accommodate which of the following substances? Okay, what does that word there, accommodate, mean? Can anyone tell me? Okay, whatever. It's just a word, right? This is English, it's not in science. So accommodate means just like, okay, it can put up with it, right? It's not that it was designated to be this, right? Like for example, let's say a shoe that fits too small. Let's take Cinderella. If you have, you can possibly kind of squeeze into a shoe that's too small, so you can accommodate your foot in there. So basically, what it's saying, like, what is something that is similar enough to things that are usually transported by this channel that can be transported by this channel, right? It's not made for this, but it can potentially do that, right? So it's asking you, what is something that's similar to sodium and hydrogen, right? Because this is a sodium hydrogen transporter. Well, what is sodium? That's Na. Typically, it's Na plus. What is hydrogen? Typically, hydrogen is presented as a proton. You're not going to have typically a hydride randomly doing this thing. Is it a hydride? I forgot the name of a nucleophilic hydrogen. I think we had this discussion last, like in the winter as well. But yes, okay. So we have things that are positive charged. Okay, let's look at anions. That's the opposite. So no cations. Yep, that's the correct answer. Phosphoric acids. Those things are huge, and they also. Well, okay, I'm not gonna draw phosphoric acid. It's really big. But yeah, there's not even a charge on this phosphoric acid right now. It's kind of just like it's neutrally charged. You know, if it's an acid, that means it has its protons. If it's a phosphorate, you know, a phosphorate means that there's just like an ion, right? It ends with 8. Typically, you're talking about an ion. Like, for example, glutamate. Glutamate is just the ion version of glutamic acid, right? Okay, so this phosphoric acid doesn't have a charge, so nah. And then neutral compounds, no, right? You're looking for something that has positive charge. B would be the correct answer for 44. Okay, any questions? If not, let us continue. Okay, here we have, oh yeah, we could, oh, okay, never mind. We'll see. Okay, let's look at 47. This one kind of got on my nerves when I took it last year because I didn't know, like I didn't know the situation. But pretty much, well, I mean, I guess I got it right. But which of the following is the correct for the compound in compound, like correct name for compound 2, right? Okay, so compound 2. What you didn't know is that if you zoomed in, they're actually dashes. They're not wedges. Snap, right? So if you got it wrong because you thought this was a wedge, I feel for you, right? Like it's really hard to determine that that's a wedge. You know, like you had to like all the way in. Is that something that happened in the double AMC? Nah, not really. Right? Yeah, everyone is just like, I can't believe it. But yes, that's what happened. But they're at least generous enough to say that, you know, the only thing that's different between these long as names is the configuration of the groups, right? So you just had to, okay, so basically it's a absolute configuration question. Bro, I thought I got it right. I was so confident. Yeah, I mean, you're confident in execution. However, it's just this, right? Okay, so does anyone want to annotate the correct answer? Like the absolute configurations, the correct answer is 2 S's. Does anyone want to do it, or should I just like explain it and then we can move on? Someone who's not confident in absolute configurations, does anyone want to do that in front of all of your peers? I feel like this definitely something TPR would do. This is to scare you to buy more products. Kevin with the economic explanation of the wedge dash phenomenon. Yeah, okay, so, oh, did someone say something?
Just said it's BS. I mean, if anyone else like wanted to do it, I mean, I could. I mean,
Yeah, go for it, Kevin.
so I kind of like knew which one was which based off of knowing that if you have, I think like the priority functional group, right, which I think would be like the ketone right here, like that's kind of like, you know, where you start off with, like, you know, which one's going to be like, you know, 3 or 4 or vice versa. But thank God in this case, like they're both kind of the same. So
make sure you articulate everything, because everyone might not be on the same page as you.
Oh yeah, so like, you know, when it comes to like numbering, you know, you always start off with like the priority functional group, like having the lowest number, and then from there you kind of just like count from there. So it'd be like, you know, 1, 2, this would be 3, and that would be 4.
Wait, my guy, this is not a carbonyl. Like the little squiggly, that's not a chiral center.
Oh yeah, no. I was just saying, like, you know, that's
Oh,
the
yeah. I was like, Kevin, no.
yeah.
Right.
But yeah, so you just go 1. This is gonna be 2 over here. I feel like I'm making it worse. Either way, it's gonna be going this way, but because of the fact that it's on a dashed, that means it's gonna have to be the opposite. So that is going to be an S configuration. Yeah.
Teaching is harder than it looks, huh? It is.
Wow.
All right, so
yeah, same thing with the other one.
yes. Ooh, I wonder if I can do this. I'm going to screenshot this, and I'm going to copy it.
Wait, I can erase my stuff though before you screen it.
Oh, fuck, you're right. Yeah, okay.
Yeah.
Go for it, Kevin. Oh, okay. So I'm gonna screenshot this, and then I'm gonna draw my box, and then I'm gonna, oh, anything pop up. Okay, whatever. So basically, if you guys can see my cursor, this group right here, we have an oxygen. Does the oxygen have a higher atomic number than a carbon group? Yep. Does it have a higher atomic number than the invisible hydrogen that it has here? Yep. How about this other carbon group? Yes. So this would be your first priority. Okay. Where everything else is pretty much a carbon group or a hydrogen. Our hydrogen is going to have to take the lowest priority, aka number 4 out of 1, 2, 3, 4. And then this carbon group, let's look at the difference between these two, right? Because if they're just carbon groups, we have to look at what they're attached to. This one is attached to an oxygen only in a single bond. This one is attached to an oxygen in a double bond. Remember, if you have a double bond, those double bonds are just going to count as 2 covalent bonds to whatever species it is double bonded to. So these are going to count as 2 oxygen groups attached to this carbon group. So this one's going to have a higher priority, right? Because this one has more oxygens than this one, right? Because the oxygen here is going to cross out. The oxygen here is going to cross out, remaining, leaving 1 oxygen species still here. So this one would be the highest priority group. So it'd be first one would be over here, second one here. The hydrogen has the 4th, and then this one would have the 3rd, right? So we go on in that direction. That would be a clockwise motion. That would be 4. However, the dash is on the OH group. The dash is not on the lowest priority group, aka the hydrogen. Thus, we must invert our absolute configuration. So first we start off with R, but because of this kind of principle that we have here, right? The dash must have, the lowest priority group must have the dash. We're just going to invert our stereochemistry from an R to an S. Same deal happens with this kind of chiral center. Okay, any questions? If not, thank you, Kevin. That's right. I appreciate you, Kevin. There's always a student who participates. Kevin's really good with it. It kind of motivates us instructors to be better. Okay, next we have 56. Okay, another freestanding one. Cool. Which of the following would be increased the most in an individual with uncontrolled diabetes? Okay, so when we have uncontrolled diabetes, what happens is just that glucose is not really able to enter your, what's it called, your cells, right? Let's say we have a blood pathway, and in diabetes, your insulin is all acting up. What does insulin do? Insulin kind of takes glucose, and insulin, it kind of like creates a receptor molecule in the cell in order for glucose to be able to enter and do its thing, right? So if you have no insulin, right, in diabetes, and you have no receptor to allow the glucose to enter, the receptor is called the GLUT4 or the GLUT2 receptor. They have different functions. I believe the GLUT4 is the main one. The GLUT2 is the one that kind of it's in the winter session passages. So you can read it over there. I don't want to mess you guys up by giving you kind of like whatever so-so information. Okay, so basically, but that's not what's important here. Basically, the glucose cannot. Oh, snap. Okay, the glucose cannot enter the cell. Thus, we're gonna have too much glucose in our blood and not enough glucose in our cell. And what does the cell need glucose for? Oh, for ATP synthesis, right? So how are we going to get our ATP if we don't have glucose? Well, remember, there are kind of different macromolecules that can donate to our glycolysis pathway, to our Krebs cycle pathway, right? And one of those things, proteins can donate, lipids can donate, nucleic acids. I've never heard of that donating for ATP synthesis. Maybe it happens, who knows? But in this case, typically we're going to be looking at lipid donation when we have diabetes, right? So for lipid donation, what we're going to have is the lipid molecule, the fatty acid molecule. Oh, it's not. Looks like so, right? It's just a bunch of hydrocarbon chains, doing their thing, and then there's a carboxylic acid group at the end. That's a fatty acid. Well, look at this. Look at these carbon groups, right? We have a 2 right here, 2 right here, 2 right here, 2 right here. Through fatty acid oxidation, we can have the creation of multiple acetyl-CoAs. Why? Because what is acetyl-CoA? Acetyl-CoA is just 2 carbon groups attached to a CoA molecule, right? And where does CoA get introduced? Can anyone tell me? Where does acetyl-CoA do its thing? Where does it shine?
The Krebs cycle.
Yeah, it shines in the Krebs. Guys, please go over your cycles, please. Yes, and guys, remember, my office hours is not just for cars or chilling. yeah, you guys can come in for tutoring too. So if you're like, oh, biochemistry, oh, like damn, biochemistry, whatever, then let me know, and then we'll like, go over the pathways, kind of like in the same depth as we did in the winter session. Okay, so yes, acetyl-CoA gets produced, right? Acetyl-CoA is just 2 carbons. So let's say we had an 8, just in just ballpark numbers, we had an 8 carbon fatty acid. So there's potential to make how many acetyl-CoAs? Can anyone tell me? If acetyl-CoA is 2 carbons and we have an 8 carbon fatty acid, how many acetyl-CoAs can we make from it?
4.
Yeah, H divided by 2, right? 4. So we can have 4 acetyl-CoAs be created from our fatty acid. Those acetyl-CoAs, good to go. They go into the Krebs, they crank out ATP, they crank out NADH and FADH2, and they go to the electron transport chain, the electron carriers, and we have ATP production through ETC, right? Perfect. However, that acetyl-CoA are not going to be things that are increased during diabetes. Why? Well, I mean, they are being produced. However, they're being used up at the same time that they're being produced, right? They're constantly being used up. So acetyl-CoA wouldn't be being synthesized. Lactic acid deals with fermentation. No one really talked about fermentation or anaerobic conditions in this question stem, so you can kindly just ignore that. The NADH is going to be used up constantly because we're talking about cellular respiration, right? So we're going to be constantly using up our NADH. That's not something that would increase. Ketone bodies, however, ketone bodies are things that can be increased. So basically, the ketone bodies is something that is going to be transporting your fat, like remember your fatty acids can donate acetyl-CoA to your Krebs cycle. The fatty acid is not going to randomly go into your blood-brain barrier and provide energy to your brain. So how is your brain going to get energy, right? Well, it's going to rely on ketone bodies. Ketone bodies are basically like just kind of what's it called? Kind of little transporters of acetyl-CoA. They can cross the blood-brain barrier. Once they get in there, they turn back into acetyl-CoA, and then they do their things. So so ketone bodies would be the one that has the greatest amount of production from all these other ones. Okay, any questions? If not, let's continue forward. All right, we're done with chemistry for the easy peasies. Any questions about chemistry before we move on? Like any strategies or something? Because remember, the basic strat to save your energy and prevent exhaustion is just to, what's it called? Read the questions first, right? All these easy peasy questions. Look, here in the beginning, I have everything that is easy peasy. That is part of my section. Look how many questions you can answer that are either full, like, are freestanding questions or attached to a passage, but you really don't need to read the passage to like answer, right? Like all these easy peasy. Like this is for chem phys, and then this is for the biochem, right? So like all these things you can catch without having to read the passage. The less peasy and the non peasy typically have passage things associated with it. Okay, so we are on biochemistry question number 2. Okay, so here it's asking us which of the following best justifies the use of DNA as opposed to RNA for generating aptamers. Okay, so I kind of have an understanding of why DNA and is always better than RNA, typically because of that OH group attached to the secondary carbon group, right? So just to give context to what I'm saying, this is your secondary carbon. If you have an RNA, you have an OH group there. If you have DNA, you don't have a OH group, right? It's a deoxyribonucleic acid, right? The oxygen group was removed. It's deoxy, right? That's where the name comes from. Deoxyribonucleic acid, DNA. RNA is just ribose nucleic acid. Why? Because it doesn't have anything changed to it. So my basic understanding is like, hey, DNA is way more stable than RNA. What's something that's an example of that? Well, our genetic information, what makes us, and our proteins and all that, what makes us who we are, is stored as DNA. It's not stored as RNA, right? If things were stored as RNA, well, that thing would be really unstable. RNA is prone to degradation. DNA is more stable for that reason. Due to this OH group right here, is this OH group. Let's say we have a phosphate group here, and then we have another just a ribose. Oh god, this is the ugliest ribose. Basically, what can happen is with if you introduce a base here, they can just cleave off like this, or actually the the phosphate would be going up like that, attaching here, and then the bottom half would be cleaving off down here. So with base hydrolysis, you can really just mess up an RNA molecule because of this basic group right here. There's a mechanism that I explained in the previous full length. The mechanism isn't really important. Just know that it happens, right? So yeah, so okay, that's my basic understanding as I go through reading through this question. But okay, let's see what an aptamer is, right? Because I have that basic understanding. So I answered this, but like, okay, what is an aptamer? Just to make sure that we're tackling it at the right angle. Because it says here, DNA aptamers are synthetic oligonucleotides that can bind specifically to target substrate molecules with high affinity. Wow, they look like little kind of missiles, right? They're kind of missiles that are going to tackle something that's important. Okay, and the aptamer are single-stranded capable of forming a tremendous variety of blah blah blah. One method of generating these molecules is called, okay, who cares? Selects. Okay, the first step of this process is generated by a large library of oligonucleotides of the same length, but with random sequences. Okay, so just a bunch of different ones. In the second step, this library is directly exposed to the target molecule bound to the surface, and then the target molecules are washed away. So there's this oligonucleotide, this aptamer goes through a bunch of stuff, right? And I just told you guys that DNA is more stable than RNA. So if RNA is going through a bunch of stuff, like if the aptamer is going through a bunch of shit, like it's being washed, it's tackling something, it's binding something, well, guess what? A small little OH group can just ruin the entire process, right? So that would be our correct answer, right? DNA is double-stranded. That doesn't really matter for the aptamer. It's single-stranded anyways. The RNA is single-stranded. That's just a random fact. That's not the reason why an aptamer should be DNA over RNA, because you can have single-stranded DNA. And then DNA lacks the 2-hydroxyl group, yes, right, because it would give it stabilization. And then the RNA contains a uracil, a uracil, Jesus Christ, a uracil instead of a thymine, but that doesn't really matter, particularly for the for our here process, because the uracil and the thymine, they're very similar to each other. The only difference between them is it's just a methyl group. So they're very similar in terms of like binding. They both bind to adenine the same way. So that doesn't really matter. It's just the fact that DNA is more stable and RNA can be degraded. Okay, any questions for 2? If not, coolio. And we will go to question 3. Okay, question 3. Same passage. Let's get it. The screening process of selects for high affinity oligonucleotides incorporates which of the following? Okay, so basically you just have to know your lab techniques, right? A size exclusion. Okay, does it state in the passage that we're separating things based off size or based off the fact that the aptamers are going to be binding to something specific, right? It's going to be the latter. It's binding to something specific. So size, NAF, right? And also, I mean, it says here that the aptamers are what? One method of generating these molecules is called blah, blah, blah. The first step in this process is to generate a large library of oligonucleotides of the same length. If they're the same length, that means that they're the same size, right? So then it's not going to be size exclusion chromatography. Also, just remember, size exclusion chromatography, it's not something that's very specific, right? It's just going to separate things based off their size. It's not, you can't, if something had the same sizes, well, how are you going to separate those two, right? Like a size exclusion wouldn't cut it. The gel electrophoresis is separating things based off size and charge, right? Just the sample is loaded to your gel electrophoresis. It goes down towards the negatively charged, actually, the positively charged terminal, and just going down that way. Things that are small can go down further. Things that are big are not going to be able to go down as much because the gel electrophoresis is a gel that have pores, and so things that are small can just like do kind of like in Connect 4 with a little like ball, like the chip goes down and goes like that. If we had a chip that didn't fit, was going to go down way slowly, if not at all, right? What in this case, the gel electrophoresis is just going to go down slowly, right? So things that are bigger. Okay, can anyone tell me which one is the bigger molecule, A or B? Damn. You see, that's why I don't want to go so fast, because I'm not sure if you guys can keep up. You guys kept up. Yes, A would be the correct answer, right? It's not going down as much. B would be a smaller thing for the gel electrophoresis, right? So now we can cross that out. That's not what's happening here. Okay, The ion exchange chromatography, again, the DNA is what? Negatively charged because of the phosphate groups on its backbone. So is there any way to differentiate between different, what's it called? DNAs using that method? No. So it's gonna be, we're gonna be using affinity chromatography, right? Where the atomer is binding to whatever it is that's specific to it. It says here in the second step, this library is directly exposed to the target molecules bound to its surface, and the target molecules are washed away so that the bound oligonucleotides remain. That's basically affinity chromatography. Another version of that would be like ELISA. If you guys know what ELISA is, that's an example of affinity chromatography using proteins rather than DNA. Okay, and antibodies. Okay, any questions for 3? If not, let's continue onward. All right, we got 22. Okay, for 22, it says here, which of the following amino acids found in the blah, blah, blah is most likely to contribute to the protein stabilization of GTP in the RF active site? Okay, so basically it's asking us which amino acid would be better. In this site, typically whenever they ask something like that, somewhere in the passage they're going to state if the site is either polar or non-polar, right? So basically we're just looking for that word. We're looking for this thing right here, and we're looking to see if it's polar or non-polar. Okay, let's look for it. Oh, here it is. Okay, let's continue. Let's see what kind of site it's got. Differentiated in curvature, transported vesicle, blah, blah, blah. These differences are thought to arise from the defects caused by formation of vesicles to result in increase in vascular volume, decrease in local, and this does have, okay, find nothing. Okay, let's continue backward. Our folate active depends on guanosine nucleotide into which is bound, either GTP or GDP, hydrolysis of GDP to GTP in the GTP A's. Activity of RF1 is dependent on the active separated, which activates the hydrolytic activation of AF1, in part by providing a catalytic residue which stabilizes the GTP binding pocket of this site. Okay, it's stabilizing the pocket with a specific residue. Where it is this residue that we're talking about. All right, let me reread the question. Sorry, guys. All right. Which of the following amino acids found in the RF-GAP1 is most likely to contribute to the protein's stabilization? Oh, I got you. My bad. Yeah, I thought it was like one of those questions, but it's really just a comprehension of what GTP is. So, guys, what is GTP? What is the overall charge of GTP? Well, first off, what is GTP? Can anyone tell me?
It's like a unit, or at least it's similar to like ATP and like how it functions.
Basically, it's a guanine nucleotide attached to 3 phosphates, right? So it's a guanine triphosphate. It's guanine attached to 3 phosphates. What's an ATP? Okay, so if that's what a GTP is, what's the name of adenine? Fuck. What's the name of ATP?
Is that adenine?
Triphosphate. Yes, that's what it is. So yeah, so guanine. GTP is guanine triphosphate and ATP is adenine triphosphate, right? Okay, so phosphate groups, what do they typically have? A positive charge or a negative charge?
Negative.
So if you want something to hold it there, remember it's going to stabilize. If you want something to hold it there and not have it like move around a bunch, we're going to want something that bonds to it very nicely, right? So let's look at our amino acids. Yeah, I just needed the question. I was wondering why this one was in the easy peasy section. Yeah, you just needed to know that your amino acid and what GTP is. Okay, let's look at the theme of our amino acids here. We have lysine, which is a base. It's going to have a positive charge at physiological pH. Valine is nonpolar. Aspartic acid is it's acidic. It's going to have a negative charge in acidic pH, and it's going to have a negative charge in physiological pH. And then we have an amide, right? It's just polar group. Okay, so which one would be best used for something that has a negative charge? Can anyone tell me? Remember, GTP has a negative charge, so what would be the thing that can hold on to it the best? A positive charge would be lysine, right? So lysine would be the correct answer for A. Sorry about that. I was tweaking. That's what happens when you don't finish reading the question. All right. So we have. Oh, we can definitely finish this. I hope. All right. Here we have question 27. All right. Julio. Can anyone find the number 27? Okay. All right, we have a freestanding question. It says the inhibition of hexokinase by glucose 6-phosphate can be best be classified as. Okay, so let's look at this reaction, right? Excuse me. Okay, so this reaction is a glucose molecule reacting with hexokinase, and ATP is going to be sacrificed, making an ADP, and we're going to have glucose 6-phosphate as a result, right? This is the first step of glycolysis. Okay, and did you see how I drew this arrow? It's irreversible, right? It only goes in one direction. The reason being is because ATP hydrolysis, man, we're not going to reverse that. Like, you can't randomly just make ATP out of nowhere. So this is an irreversible reaction because for one, that fact, and two, for the fact that it's just so thermodynamically spontaneous, right? Just like this ATP breaking apart, this releasing so much energy, that's not reversible. You can't randomly just like take back energy and squeeze it. So this is going to be an irreversible step. Glucose 6-phosphate can continue onward into the glycolysis pathway and straight up do its thing and make ATP, or it can be shuttled out and go into the pentose phosphate pathway, right? There's the two possibilities of glucose 6-phosphate, right? However, oh, and just a quick side note, once a phosphate group is attached to glucose, it can't leave the cell, right? The glucose on its own without a phosphate group, it can just go in and out of the cell, do its thing, could be transported, who cares? Glucose 6-phosphate can't do that. Can anyone tell me why glucose 6-phosphate can't move around in and out of the cell like glucose can?
Is it because it's like fixed in a certain way? Right?
Mm,
What was that?
really? What's the difference between glucose and glucose 6-phosphate? The phosphate.
Phosphate, yeah.
What does a phosphate group have?
Oh, the negative charge.
Can the negative charge go through that non-polar fatty the membrane? Remember, the membrane is a hydrophilic head and then hydrophobic tails. Things that are non-polar, like benzene, can just go through those hydrophobic tails. Who cares? Lickety split. Something that's charged like glucose 6-phosphate won't be able to. So yeah, so once the phosphate is attached to your glucose, it's trapped in there, right? So we want to make sure that we're using this kind of process. We want to make sure we're not using it haphazardly, right? Because if we were to use it haphazardly, one would be wasting a bunch of ATP. Two, the glucose would be just trapped into a cell. We don't want our glucose trapped in a cell. What if a different cell needs that glucose, right? So there's a lot of reasons why we want to regulate this step. Well, what's the fastest way to regulate this step? With a negative feedback inhibition, right? If there's a bunch of glucose 6-phosphate and they're just like kind of lying around, and glycolysis is like it's already full speed, like it doesn't matter how much glucose 6-phosphate you have left, right? Like it's not going to go any faster. And if the pentose phosphate pathway is nice and taken up, like there's no need for it, or if it's already acting at full speed, and there's a bunch of glucose 6-phosphate, well, guess what? Let's say this is like the button right here. This is the button to like stop hexokinase. Well, if there's just a small amount of glucose 6-phosphate, are they going and they're like kind of floating around? The chances of them hitting that button are low, right? But if you have a bunch of glucose 6-phosphate everywhere, because there's an abundance, well, guess what? The chances of this thing being inhibited is really high. So whenever you have a bunch of glucose 6-phosphate, what's going to happen is that it's going to bind to your hexokinase and stop it, right? Because it's just like, hey, man, we have enough. Like, I am what you make, and there's too much of me. We're wasting a bunch of ATP. The glucose is being trapped into the cell. No bueno. Stop it, right? So that's feedback inhibition, right? That's basically what feedback inhibition does. The product of the enzyme is going back and inhibiting the enzyme. So basically, this goes from here, smacks there, and it stops the process, right? That's feedback inhibition. Happens quite often in cellular respiration. Proteolytic cleavage. No, if you cleaved, remember, an enzyme is what? It's a protein. If you were to cut your enzyme, you would ruin the structure. Can anyone tell me what gives a protein its function?
Oh, structure.
The structure gives its function, right? Because the primary structure of a protein is just its chain of amino acids, right? Blah, blah, blah. And then it folds based off the hydrogen bonding, and then it does its more stuff based off other things like hydrophobic and hydrophilic interactions. So its structure is what gives it its function. So cool. You're not going to be randomly cutting your enzyme. That's completely knocking it out of contention. Like it loses its function. So that's not what's going to happen. Enzymatic catalysis. Yes, enzymes do catalyze things, but that's not inhibition, right? That's just randomly saying enzyme catalysis. And then positive feedback is the opposite of negative feedback. Positive feedback is just saying like, hey man, I like the way you're making this product. Basically the product is going to go back and hit the enzyme, telling it to make more of itself. That's positive feedback. So instead of inhibiting it, like in this case, it could be like, no, stop it. Let's say just enzyme or reactant product. The product is just like, hey man, good work. Keep doing what you're doing. That's positive feedback. Right? Okay, any questions? Nope. Okay, moving onward. Guys, please don't slack on your pathways. So the easiest things to memorize. Why? Because everything is conveniently named in biochemistry, like for real. Like hexokinase, something that adds a phosphate group, a kinase, to a hexose, a 6-carbon structure. Like that's like, it doesn't get better than that, honestly. All right. So we got question 28. Okay, during times of starvation, like currently right now, what's happening with me? What happens? Okay, triglyceride stores in adipocytes are anabolized by lipase. Okay, well, I'm not sure about this whole triglyceride stores thing, but if you look at it over here, the anabolized by lipase isn't anabolize thing. Anabolize is the building up of structures. Lipases and proteases and alpha amylases, those things cut whatever it is that they're assigned to. So that's catalysis. So cutting something and making it smaller and breaking it down is catalysis. So this anabolization is really just going to go down. A triacylglyceride. Guys, can anyone tell me what a triacylglyceride is? Well, let's look at the name, right? It's a glyceride. And glyceride is this bad boy right here. OH, OH, OH. If we have a triacylglyceride, well, basically what happens is that those hydrogens are plopped off and we're going to have acyl groups on them instead. Oh, God. We have acyl groups on them instead. An acyl group is this. Can anyone tell me what this looks like?
It's just a fatty acid.
No, it's not a fatty acid. So,
Lipid.
yeah, it's a triglyceride.
Oh, yeah.
Yeah, fatty acid is just one of these guys.
Oh, my bad. Yeah. Yeah.
Mm-hmm. Having 3 of them attached to a glyceride to a glycerol group is a triglyceride. Triglyceride are stored in adipocytes for basically energy. You know, let's say remember in the starved state, for example, right? We don't have any glucose in the blood. So what we said is we can take the fatty acids, cut them down into acetyl-CoAs and make energy. So cool. But the reason why this is wrong is because of the anabolized part, right? You don't anabolize with lipase. You break things down with lipase. Okay. Let's look at B. Fatty acids are broken down via oxidation. Okay, that's something that happens. Yep. Hydration and cleavage reactions to generate NADPH and oxidize FADH2. Okay, that's where you lost me, right? The NADPH is first off not generated in fatty acid oxidation. Can anyone tell me where it is generated?
In synthesis.
Yeah, where's ADPH generated?
Oh, well, there's the pentose phosphate pathway that's
Yes.
made.
Mm-hmm. And there's another obscure pathway that I'm not sure you guys will need to know. So I'm not. Yes. And what's it called? And FADH, it's not oxidized. It's if anything, FADH is something that's being produced, so it's not going to assess wrong on 2 counts, right? For C, glycerol is converted to DHAP. So like DHAP is one of the intermediates of the glycolytic pathway. It's something that glycerol can turn into DHAP. So okay, that part is correct. Glycose, yep, it is an intermediate, which is then used to generate lactate and ethanol via fermentation. Okay, what's wrong with this last part right here? This is something that's half right, half wrong.
Can't it only make one of them? Like it can't make lactate and ethanol?
Mm-hmm. As humans, we have lactic acid fermentation. Yeast cells, for example, create ethanol fermentation. That's why you use yeast whenever you make beer or alcohol, right? You just ferment yeast and they make ethanol, our savory molecule. But also, where in the question stem did it say that we're under a state that needs fermentation in the first place? Just because you're starved doesn't mean that you have oxygen. If anything, when you're starved, that's when you have more oxygen. You're just like, I'm hungry, right? So you're going to have plenty of oxygen. So you're not going to have any fermentation. Okay, so let's look at D. Fatty acids are transported in the blood via binding to blood proteins like albumin. Yep, albumin does have a bunch of albumin is your transport protein. If you ever need something to be transported in the blood, you're most likely going to be transporting it through albumin. Okay, and then they are broken down via beta oxidation in target tissues. Yes, that's basically what fatty acid oxidation is, right? It's the transport of triglycerides from adipocytes with albumin. First, they break down the triglycerides in the adipocyte, and then the fatty acids, individual fatty acids, are transported through the albumin, and they go into their little cell, whatever that needs it, and then they break up their fatty acids into 2 carbon groups, making acetyl-CoA. Okay, any questions for 28? Okay, if not, let us move on to 34. And I'm not sure if we can get all the questions, but we'll try to do as many as we can. or we can leave these last 10 minutes for any questions. Do you guys have any questions pertaining
I think I have a question about the one before.
the
So,
28?
Like albumin.
Yes.
Like what about chylomicrons then? Like, do chylomicrons contain albumin, or is it like 2 totally different things?
So chylomicrons, that's more biology, as Ashane, but chylomicrons are found within the digestive tract. The chylomicrons are the things that are going to be holding onto your onto your digestive fats. And I believe your chylomicrons are going to be transporting those fats into your adipocytes. I studied this last year. I don't really remember all the specifics of digestion, but yeah, your chylomicrons are more part of the digestive tract, and the albumin is going to be more for the use of metabolic pathways.
Got you. Thank you.
Mm-hmm. Yep, no problem. Because the chylomicron, if you Kevin, do you know how big a chylomicron is?
It's like pretty small, right? It's low density lipoprotein. I don't know.
That bitch huge, man.
Oh.
Chylomicrons are big. So like, yeah, so that's why like you need to like absorb a bunch of fats in your, let me just pull up a picture of a chylomicron. Can anyone tell me how that thing's spelled?
CHYLOOMICRON.
MICRON. Shut up. No, not videos. Joy micron. Yeah, them bitches huge. So yeah, so basically a chylomicron, it's got a bunch of fatty acids doing its thing. You're not going to be shooting this into a cell. Like the cell would be overwhelmed if you had this many fatty acids introduced. And albumin, let's see what an albumin looks like. If you guys were wondering where I lost my grades for my GPA, it's because of English. I'm just horrible with English and spelling and in general. Yeah, this is an albumin, just a protein, and it does its thing, right? It's a really small thing. A fatty acid comes and binds to it, and it transports it. Okay, but yeah, the chylomicron is more of a digestive thing that I remember from when I studied this last year. Okay. Does that work?
Yeah, thank you.
Cool. Any other questions? I'm a bio major. I know things. Okay, that was cringy. All right, let's move on to 34. But I do know things. Guys, please remember to not slack on your psych soc. Really, if you're getting anything below on your actual MCAT, if you're getting anything below 128 on psych soc, you're just playing yourself at that point. All right, because it's just a big vocabulary quiz. All right, so 34. Let us look. N-word. No. Usually, whenever I can't say a word, I just say the N and I say the word. Okay, but you can't say it for no. All right, so this neverprine is an example of a non-nucleotide reverse transcriptase inhibitor. Which of the following is true of neverprine? All right, so if it says that it's okay, so basically we have to look into the passage for what neverprine is. Okay, so what neverprine is, let me just do a little of that. Wait, what? It's not even here. Okay, so let's look into our passage. Neverprine is an example of a non-nucleoside reverse transcriptase inhibitor. Okay, so let's look for non-nucleotide reverse transcriptase inhibitor. Right, so okay, the human HIV, blah 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 envelope is studied with 2 main glycoproteins, this and that. Okay, I'm looking for an inhibitor. La di da. Came from host cell. After reverse transcriptase, the DNA is integrated into the host genome through the use of viral enzyme integrase. Okay, after the reverse transcription, the blah, blah, blah. Basically, you're scanning for inhibition. Second paragraph in the middle. Thanks, Yannicka. All right, so AZT, oh, I've heard of AZT. Is a thymo analog. Oh, that's what AZT does? Smart. Okay, AZT is a thymidine analog that is lacking a 3-hydroxyl group necessary to end the elongation of growing DNA. Isn't that nuts? Basically, if you have a cancer cell, right, it's going to be replicating its DNA. If you throw in some AZT in there, you can't replicate the DNA. Like that. People are smart. Because ACT is a, okay, blah, blah. Crazy, right? I know this one in Spanish. Local. All right. All right. Yes. So, I mean, we have 4 minutes. Okay, 6 minutes. This would result in the arrest. Okay. So basically, okay, where's the inhibitor part? This cellular death is responsible for some of the side effects. Blah blah blah. Namely, okay, who cares? Okay, it's a non-nucleoside. Okay, there you go. A non-nucleoside reverse transcriptase inhibitor because it binds to an allosteric site on reverse transcriptase. Perfect. We have the information we need. It's an allosteric inhibitor. Okay. What is something that is an allosteric inhibitor? What is it called? It's called a non-competitive inhibitor. That's basically it. There's different kinds of inhibition that you can have. You can have active inhibition, which is, you can have competitive inhibition, which is when your inhibitor binds to the active site of the enzyme, basically occupying the space so the substrate can bind. You have non-competitive inhibitor, which is, what are we talking about over here? The non-nucleoside inhibitor, in which our nucleoside inhibitor kind of binds to an allosteric site. An allosteric site just means a site that is not the active site, right? It's just a site that's beyond it. It could be behind the active site, right? Whatever it is, just not the active site. It binds to it, and it changes the configuration of the active site so that the substrate can't, like the enzyme can't catalyze the substrate, right? So it's just like, okay, basically it inhibited, and it had to compete for the active site because a competitive inhibitor has to compete for the active site with the substrate. In this case, the inhibitor has no competition. Why? Because the substrate is not going to be binding to this allosteric site. Only the inhibitor is going to be. And then your uncompetitive inhibitor is when you have the enzyme substrate complex. So it's doing its thing. The process is happening, but the uncompetitive inhibitor comes in and binds at that moment, causing it to like not complete the reaction, right? So there's 3 types of inhibitors. When you have an allosteric inhibitor, you have a non-competitive inhibitor. Okay, any questions for 34? And we could just leave it there because unless 35 or whatever question is next is super easy. 43. Any questions for 34?
Okay, Brian.
Yes.
Brian, can you? I have a question about 34.
Yes.
I thought I remember uncompetitive inhibition being allosteric too.
Yeah, it can. So basically, what the uncompetitive does is it attaches to the enzyme substrate complex and prevents the enzyme from like doing its thing. In this case, the picture has it, what's it called, affecting the active site. It can also bind to a different part. Basically, the uncompetitive inhibitor is kind of like an umbrella term. And the umbrella term is that once the enzyme substrate complex is formed, the uncompetitive inhibitor kind of stops it from happening. The non-competitive inhibitor and the competitive inhibitor happen before the substrate is bound to the enzyme.
Got it. Thank you.
I thought uncompetitive inhibitor is allosteric binding too. Yes. Oh, snap.
I have a quick question.
Uh-huh.
So is it it's competitive inhibition where if you add more of the concentration of the substrate, you're able to overcome the inhibition effect. But can you do the same thing with non and un?
For the non-competitive, not really, because the substrate is not really kind of like being like its ability to like go there is like that's why it's non-competitive. And the uncompetitive either, like the substrate is able to bind, it's able to do its thing, but just once the uncompetitive inhibitor comes in and binds, it kind of screws it over and it doesn't let it do its thing. Same thing over here. With the terms of the concentration of the substrate, if the substrate is in higher concentration than the inhibitor, then there's going to be less inhibition, but that doesn't mean that there isn't going to be an inhibition overall.
So for uncompetitive and non-competitive, can you ever like kind of compensate for the inhibition or no?
Yeah, if you add more enzyme.
Okay, gotcha, thanks.
Okay. So just to address Vinny's thing. It's 9:59. Yes, we'll do enzymes and all the nit and grit about the inhibition and all that in the content review of next week's Saturday. Okay, guys, I'll see you then, and by the time that happens, I will have a beard again, hopefully. Okay, everyone.
Thanks, bro.
Really quick
Adios.
question. Can you just like quickly explain? Like, I always get confused with what mixed inhibition is. Like, I know the other ones, but like, do you know what like mixed is?
Yeah, mixed inhibition is tricky because it doesn't have a defined like kind of route. Like it doesn't have a defined because like you can mix inhibitors is basically kind of like the umbrella term for things that are not categorized as competitive or non-competitive or uncompetitive. So it's just a bunch of different things that can do different possibilities, and that's why the Km can vary, the Vmax can vary if you know that. So we'll go into depth in that next week. But yeah, basically just know for mixed inhibitors that they can affect the Km and the Vmax differently depending on the mixed inhibitor. They'll tell you basically if they give you a mixed inhibitor, they'll have to show you the Lineweaver-Burke plot
Right.
to show you like whatever it's affecting.
Okay, thank you.
No problem. All right, bye everyone.